becomes relevant. Oh, should I start again with the recording?
Oh, I'm starting to record. This is fine.
Okay, yeah. So to give you a structure of how the talk's going to go, I'll start by giving some background and motivation. Then I'll introduce ProGen3, which is a family of sparse generative protein language models, and we'll get into what all of that means as it becomes relevant. After that, since these are generative models, I'll present both a computational analysis as well as an experimental analysis with lab data of the proteins that are generated by the ProGen3 models. After that, I'll talk about how we can use a limited amount of laboratory data to align ProGen3 with notions of protein fitness that we as protein designers might care about. And then finally, I'll wrap up and talk about some next steps. Logistically, feel free to interrupt at any time with questions. I think, yeah, I won't be able to view the chat, so TBD on what exactly works best, but maybe if Bo, you could relay any questions that come up in the chat or something like this, I'm happy to answer them as they come up.
Yeah, cool. I guess we can also have like a Q&A section in the end, and we would then at that time we'll go through the question in the chat.
Sure. Yeah, but happy to answer those live, so let me know as they come up.
Okay.
Awesome. So let's get into it. I'm sure I don't really need to convince many of you of this fact, but let's just start briefly by talking about why protein language models actually matter. So at this point, PLMs have accelerated design for many important protein families, including gene editors and antibodies. And these proteins have applications in a wide range of fields that include therapeutics, agriculture, biomanufacturing, just to name a few. To be a bit more concrete, we at Profluent have used the previous generation of ProGen models, the ProGen2 models, to design highly active gene editing proteins, specifically Cas9s and base editors, that we found had higher activity and higher specificity compared to some of the industry standard proteins, some of which were derived through like 8+ rounds of directed evolution. So now, talking a bit about how these PLMs actually work, for this talk, I'm going to be focusing on sequence-based transformer protein language models. And how these models work is they take as input a sequence of amino acids, they'll embed it, and then they'll push it through a bunch of transformer layers before doing some sort of a residue prediction task. And in each of these transformer layers, you have two components, which I'll touch a bit more on later. These are the self-attention component and then a feed-forward neural network. So the way you determine a protein language model's capabilities are really based on these two features, the self-attention and the residue prediction. So for example, if we're dealing with autoregressive models, the residue prediction task is to predict the identity of the next amino acid at every position. And the way you achieve this is through this so-called decoder self-attention, where each token attends to all of the tokens that came before it, but not after. And in this way, that means that you're predicting the next token conditioned on the prefix, but not the suffix. So in this way, you can use these autoregressive models to generate new proteins. Conversely, with a masked language model, what you'll do is you'll take some of these tokens and you'll replace them with a mask. And then the goal is to predict the identity of the masked token. In order to do this, you have this encoder-style bidirectional attention, where every token attends to every other token. And in this way, you get a context-sensitive representation of each position that accounts for the full protein context, both in front and behind it. And so this kind of sets up like what these different architectures are actually good for. So as I alluded to, autoregressive or causal PLMs can generate new proteins, and this can be either from scratch, conditioned on a prefix of the protein, or even conditioned on some external information like structure. And they can assign likelihoods to natural generated or engineered proteins. And the community has found that these likelihoods often correlate well with experimental measurements of protein fitness that we care about and that we might want to optimize. Conversely, masked protein language models are really good at learning representations of proteins that enable the prediction of protein properties and even three-dimensional structure. They can't exactly be used for generation, but what they can do is propose a small number of mutations to an existing protein. And they can also assign these pseudo likelihoods to proteins, and those likelihoods often correlate well with protein fitness. Now, oh, sorry, was there a question? Okay, yeah. So maybe now taking a step back, in NLP, one of the most common ways that people use to improve these models is through scaling up both the is through. Oh,
I think
what's going
the
on?
slide is gone.
Okay. Let's try that again. Um,
Would
uh,
that help if I make you to co-host?
um, sure. Yeah. Let me try sharing again. Um, all right.
Oh, sorry, it's fine.
Okay. Are my slides back up?
Yeah,
Okay.
it's good now.
Cool. Yeah, so in NLP, one of the standard ways that people improve these language models is by scaling them up, both in terms of the model size and the amount of data that goes into those models. And so in terms of proteins, uh, it's a natural question to ask, like, what scaling up really buys you? And this study by Cheng et al., which I'll reference a couple times throughout this presentation, found that as your models get bigger, they get better at capturing the natural distribution of proteins. And so in this plot on the horizontal axis, you have the amount of compute being used to train the model. And on the vertical axis, you have its validation loss. Basically, how well that model understands the evolutionary distribution. And based on like training a bunch of different models for different compute budgets and varying basically both the amount of data and the model size, you can derive these compute optimal scaling laws, which tell you for a fixed compute budget, what's the best way in which you should divide up your model size versus dataset size. However, there's this nice Substack post from Pascal Notin, which showed that for a wide range of different protein language model families that have all sorts of different architectures and functions, it's a pretty consistent finding that once you scale beyond a certain point, their fitness prediction performance actually starts to degrade. And this is in terms of like measuring how those zero-shot likelihoods correlate with experimentally measured protein fitness. And so this suggests some sort of a fundamental tension between this task of better learning the evolutionary distribution of proteins and predicting properties that we might care about. And so with that, this brings us to some of these open questions for generative protein language models specifically. The first is, what's the optimal data distribution for pre-training? This question has been pretty understudied in the literature, and in NLP, it's been found that the specific pre-training data can make a pretty big difference to the final model performance. Second, what's the optimal trade-off between model size and dataset size? This Cheng et al. Study does address this question, but it's pretty unclear whether their findings generalize to new training distributions or model architectures, given that these factors have had an impact on scaling behavior in NLP. Third, how does model scaling affect the proteins that are actually generated by these PLMs? And moreover, do those findings hold up in the wet lab? And then finally, how can we use laboratory data to align generative protein language models with a specific property of interest? And does model scale play a role in this? And really, the goal of this alignment step is to see, like in a zero-shot sense, these models, once you scale above a certain point, like their zero-shot fitness prediction performance starts to degrade. And so the question is, are the representations these larger models learn, are they more useful for these sorts of tasks? So you can, with a limited amount of data, get better performance on optimizing these properties. Any questions at this point? Awesome. So yeah, I guess with that, I'm gonna talk about the actual ProGen3 models. So ProGen3 is a family of autoregressive autoregressive language models. And the key modifications we make, and we make two key modifications to the standard transformer architecture to achieve all of its capabilities, both to the self-attention as well as to the mixture of experts. For the self-attention, we still have this left to right attention mask, and that by default enables you to do protein generation from the N terminal to the C terminal, or from the C terminal to the N terminal. One of the new capabilities we add in ProGen3 is also this ability to fill in the middle or do infilling. And the way we achieve that is we have this sentinel token in the middle, which replaces a full span. And then once you have the full sequence, both the prefix and the suffix of that span, you'll repeat that sentinel token, and then you'll generate that span one token at a time. So to give you a bit more of a concrete example of what this looks like, let's say that we're interested in SPCas9, which has this sequence, and we want to infill the magenta span. So the first thing we do is we pull out that magenta span and replace it with this span 0 sentinel token. And then once we put that span 0 token at the end of the sequence, we will just generate the relevant span one token at a time autoregressively. The next component that we modify is the feed-forward network of ProGen3, and we replace it with what's called a sparse mixture of experts. So the way this works is that each of these, in a single MOE layer, we have a bunch of different experts, each of which is just an FFN. And so practically speaking, we might have like 8 different experts in this network. And the way these MOE networks buy you huge efficiency gains is that instead of activating like all 8 of your experts the way you would with a dense model, you instead push your embeddings for each token through a router, and that router will selectively choose just the top 2 experts from this ensemble of 8. And in that way, you're only activating 2 out of the 8 experts for each of these inputs. So this sparsity gives you a big efficiency boost since you're only using like 25% of the compute. In actuality, it improves your throughput by a factor of 3 just because like there's some inefficiencies in these sparse implementations, but it's still a pretty big increase in throughput. But a pretty big question that arises is, do you actually like see the same model performance from these sparse architectures? So to answer this question, we took a dense architecture and a 25% sparse architecture, basically activating 2 out of 8 experts, as I mentioned before. We fixed the amount of parameters and we fixed the amount of training tokens. And then we evaluated the validation loss at the end of training. So you can see that for a fixed parameter count, you get a slightly worse validation loss with a sparse architecture. However, you should notice that you're using about 3 times fewer flops. So if instead we take this 1.3 billion parameter model and scale it up to 3 billion parameters, we're still using about half the flops as our dense model, but we get a much lower val loss. And so that suggests that sparsity is basically gives you more or less free efficiency boosts gives you more or less a free efficiency boost to your model and lets you scale a lot bigger than you might otherwise have with a dense model. Now that we've settled on an architecture for this model, we need to talk about the data that actually went in this. And in order to train the ProGen3 models, we assembled the Profluent Protein Atlas, or PPA, from more than 10 different genomic and metagenomic sources. And we applied some pretty rigorous quality filters, including removing protein fragments. This is something that like historically we found can make a pretty big difference to model performance. And to our knowledge, pretty much only the OMG corpus of like these other big ones out there does this step. And the PPA version 1, which is what we trained ProGen3 on, is on par with the largest assembled protein resources, and we are continually expanding it. So for instance, PPA version 3 is 3 times larger and has 10 billion full-length protein sequences in it. With this massive amount of data in hand, an important question is, well, what's the data distribution that you should pre-train on? And this is a question that's been pretty underexplored in the literature. And the axis we chose to focus on is the diversity of the data. So to give you maybe an illustrative example, let's say that we've clustered all of our 3 billion proteins at 50% identity. You can consider a few different ways of sampling that data. So the simplest thing you could do is just to take the natural distribution, what I call unmodified, as is, and you make no modifications. On the other extreme, you could uniformly sample all of your 50% identity clusters. So basically, if a cluster has size 1 versus a size a million, you'll sample them with equal probability. You won't take into account like the actual size of the cluster. And then you have these two options in the middle, like square root and inverse log. And you can kind of see like they do a good job of covering like the overall diversity space. On the horizontal axis here is just the cluster size, and then this is a cumulative density accumulating like how many of the protein sequences fall into each of these clusters from each of these distributions. So what we find here, so in order to evaluate, we construct 3 different val sets. One of them is in distribution, distinct from the training data at 90% identity, and 2 are out of distribution. They're distinct at 30 and 50% identity, respectively. What we find is that inverse log achieves the best balance of in distribution and out of distribution performance. And quite surprisingly, this uniform distribution performs by far the worst across the board. And I think this is notable because a lot of the existing protein language model works tend to bias towards a more uniform distribution. So for instance, if you're just training on UNIREF 50, this is equivalent to training on a uniform distribution. And at least for autoregressive models, perhaps the story is different for encoder models, it seems pretty clear that training on a uniform distribution really hurts your final performance. And we find that this inverse log distribution is the right middle ground here. And maybe for a bit of intuition as to why this might be happening, if you think about like with a uniform distribution, you'll have a lot of these really tiny clusters, like let's say 1 or 2 or 3 proteins in them, which you're sampling over and over and over again. And so the model will probably learn some really weird overfitting modes to those clusters to those sequences really that it's seen a bunch of times, whereas it might not get as full a picture of some of these better represented clusters in the data. And so basically with all of these hyperparameters settled, we started scaling up. And to do that, we trained 37 different models ranging in size from 112 million to 10 billion parameters on data sets that range from 10 billion to 1 trillion tokens. And you can see for all of these different model sizes and data set sizes, we plot their final validation loss. And what we do is we fit a scaling law that basically predicts the final val loss as a function of the model size and the dataset size. And we find this relationship that the optimal number of parameters scales as something like the dataset size to the 1.479. And based on this, we can derive like what's the compute optimal allocation for 10 to the 23 flops. And based on that, we decide to train this 46 billion parameter model on 1.5 trillion tokens from the Profluent Protein Atlas. And one note I'll make is that our finding here with the specific scaling laws we get largely agree with the Chang et al. Paper I mentioned previously, which suggests that this is a pretty general finding for autoregressive protein language models, that your model size is more important than your dataset size. And that's contradictory to the prevailing wisdom for NLP, where the two are weighted equally to each other. Yeah, happy to take any questions at this point. Right. So moving on to the next section, let's talk about how we like generating proteins from these models and what they look like. So the first sort of computational check we can do is to see how protein-like some of these generations look according to a variety of bioinformatic filters. And so to give you a sense of what protein-like actually means, first, we know that language models in general are known to generate really repetitive sequences. And so we remove sequences that consist of more than 25% repeats. Specifically, we use Tintin and remove all the sequences with more than 25% low complexity regions. Next, if a generation doesn't have an end of sequence token, it's either an artifact or an incomplete generation. So we remove that as well. And then finally, most proteins should probably have at least 80% alignment coverage to some other known protein, even if the percent identity is low. And so these are the three filters we apply to determine like how protein-like something is. And across the board for the ProGen3 models, we find that the larger models have more sequences that pass each of these filters. And it's especially notable that ProGen3 46 billion generates far fewer repeats than ProGen3 339 million, suggesting that's a major improvement. I'm also comparing here to ProGen2 extra large, which is a previous generation model that was 6.4 billion parameters and trained on about 350 billion tokens. So kind of comparable in scope to ProGen3 3 billion. But we see that this model has just much worse performance in terms of generating valid sequences. And I think this really highlights the importance of some of the architecture improvements and data distribution improvements that went into the ProGen3 work. Was there a question?
Oh, there's a question in the chat. How is the scaling law different from the natural language?
The scaling laws. So here you'll see that there's this like optimal number of parameters scales as like the data set size to the 1.47. So that suggests that you should scale your model size faster than your data set size. In NLP, the scaling laws have generally shown that that these two should scale in lockstep, that this exponent is just one.
Okay, got you.
Yep. Cool. And then in terms of the diversity of these generations coming out of ProGen3, the highest level takeaway is that larger models cover more of the natural protein space. So this is a bit of a busy figure, but I'll go into it panel by panel. So the first thing we do is for all of the valid generations that we were just talking about on the previous slide, we search them against the entire PPA, and we see how many of the clusters of the 30% identity clusters in the PPA get covered by these model generations. And basically here we plot like based on how many proteins you generate, how much of these natural clusters do you end up covering? And we find that as you scale up the ProGen3 models, you cover more and more of these naturally occurring clusters. And even amongst its natural generations, you'll see that ProGen2 XL generates less diversity than the smaller ProGen3 3 billion parameter model, while also being a lot less efficient. And the next thing we do is to see how do these clusters that each of these models generate from, how do they overlap with each other? And what we see is that the clusters covered by the larger models are mostly a superset of the clusters that are covered by the smaller models. So as you're scaling up, you're not really losing capabilities. You're just gaining the ability to cover more of protein space. And mechanistically, the clearest reason we saw as to like why these larger models are covering more of protein space is that they're better able to represent rarer clusters. So here on the right panel, we take the sequences from each of the models and we map them onto the size of the clusters that those sequences get mapped onto. And you see that the larger models are much more likely to generate from these really small, rare clusters, which the larger models are unable, or which the smaller models are unable to generate from. All right, I'm gonna start talking about like the experimental evaluation. If there's any questions on this part, like let me know, but for now I'll move on. So in terms of how you evaluate these general protein language models in the lab, the assays have some pretty difficult requirements. First, we need something that's agnostic to protein family and function. Second, we need to measure a pretty general concept of protein fitness, given that we have potentially a bunch of different like families and functions. Third, it needs to be amenable to high throughput operations because, as I'll talk about in a bit, we test more than 1,000 different proteins for this assay. And this pretty much leaves us with two options for what we measure. The first is thermostability, and the second is soluble protein abundance. We end up going with soluble protein abundance just because it ends up measuring a much more general notion of protein fitness compared to thermostability, which is a lot more specific. And with that, I'm going to introduce how the assay actually works in a very simplified way, but hopefully this gets the key ideas across. So what you do is, so maybe for a bit of background on GFP, green fluorescent protein, this protein is built of a beta barrel that has 11 different beta sheets that are all kind of like in this barrel structure. And for this assay, what you do is you fuse one of the sheets, one of these 11 sheets, to your protein of interest. And then separately, you express the other 10 sheets all fused together. And the idea is if your protein of interest expresses, then these two pieces will fuse together and you'll see fluorescence. We really like this assay because it indirectly measures a lot of quantities that we care about. It measures protein expression. It measures certain notions of like stability and foldability, since you need to make sure that your protein of interest isn't just like aggregating all together. So the like this GFP1 thing is actually accessible to like GFP2 through 11. And it also assesses the toxicity of the thing that you're creating to E. coli in this case, because if the protein is toxic, then you won't see fluorescence because the cells will just die. And now going into the way we actually designed the experiment, as I mentioned before, the generations fall broadly into three groups of clusters. So first, we have 42 clusters that are covered by all three of the models. Then we have 62 clusters that are covered by the 46 billion and 3 billion parameter model, the two larger ones. And then we have 45 clusters that are covered by just the largest model. And from each of these clusters, we select two natural proteins. These will essentially be our baseline, our control. Then we select one generation per model that is 60 to 80% identity to a natural sequence. This is the quote unquote easy generation task. And then we select one generation that's 40 to 60% identity to a natural. And this is the harder out of distribution task that we set up. So all told, as I mentioned, we tested more than 1,000 proteins. And the overall finding here is that for all of these different buckets we constructed, a similar fraction of proteins, like across the board, the total levels of expression were actually really similar for all three of the models in all 6 of these generation modes. And basically this dot that you see on each of these plots is the amount of expression that you got for the naturals in those clusters. And so you'll see that the amount of expression you get like across the board from all of these models is really similar to the expression level from the natural proteins. However, the larger models are able to generate these expressing proteins for much wider swaths of protein space, especially, as I mentioned before, for some of these rarer protein families. And so that suggests that, like, if you're comparing these models head to head, there may not be huge differences, but the advantages of scale really lie in the ability of these models to capture more of the overall protein distribution, especially for families that might not be super well represented. And to show you in action, like like some of the implications of this, we found that larger models are generally better at infilling. So we pre-selected 18 natural proteins, and we infilled both short and long spans. And we found, so in this plot, on the horizontal axis is the relative expression of the generations from the 3 billion parameter model. And this is relative to the natural protein that was used as the seed for generation. And on the vertical axis is the relative expression of the 46 billion parameter model's generations. So anything in the anything in the top left is a infilling task where the larger model outperformed the smaller one, and the lower right half is where the smaller model outperformed the larger one. And we see for the short infills pretty clearly that the larger models tend to achieve higher expression. For the longer infills, it seems like this was just a hard task, and so it all kind of looks like a wash. And the reason that we see this is that more proteins of these pre-selected naturals were in distribution for the larger model, but not the smaller model. And we never saw a case where something was in distribution for the 3 billion parameter model, but out of distribution for the 46 billion parameter model. Yep. Any questions before I move on to talking about alignment?
Sorry, there are a bunch of questions in the chat. You want to go through it now, or we should like immediately get in?
Yeah, let's go through them.
Okay. The first question is for the slide that like different model side like cover different number of clusters. Are these all unconditional generations?
For this slide?
Yeah.
Yeah, these are all unconditional generations.
Oh, okay, nice. Leo also asked question, how do you define repeated in protein sequences?
A repeat?
Yeah.
This is based on Tantan. So Tantan is a tool that will go through your protein and identify like regions that it determines are low complexity. I'm not 100% familiar with like the exact algorithm they use, but like, for instance, if you have like I don't know, like a bunch of alanines in a row, or like you have some like GS kind of thing. Like those are examples of like low complexity regions. And so that's basically saying that, so if more than 25% of your protein consists of these low complexity regions, then it's considered to be too repetitive and is removed. And for context, I think something like 98% of the PPA passes that filter. So it's a pretty generous.
Okay. You'll also ask what about the generation below 40% ID?
Yeah, for the generations below 40% identity, I don't have those in this presentation. There's more details on those in the paper itself, but I think TLDR there is that the larger models maybe do a little bit better at those generations, but the differences aren't huge. One thing that is cool, though, is all three of these models are able to generate like expressing proteins that are less than 30% identity to any known natural.
Okay. Another question, could you explain more about the experimentations? How does express 1,000 of proteins? It is conventional for E. coli expressions or cell-free protein expression?
Yeah, so I'll do my best. I didn't do the experiment myself, but this was a big arrayed screen. Each well had like one protein. There was a lot of use of lab automation to make stuff work at that scale. Uh, we tried like a few different versions of the assay. What ended up working best was measuring the cell lysate. Um, there's a lot of details in the paper itself, though. In one of the appendices goes into a lot of detail into the exact experimental protocols.
Okay. The last question we have in the chat. For the figures showing percentage of protein expressed at each bin of sequence identity to natural sequences, how were the clusters selected, and were there specific classes where ProGen could or could not generate?
Yeah, so just due to experimental bandwidth, we didn't actually do any experiments with ProGen2. We focused on ProGen3, we focused on ProGen3 for like the actual experiment. And I think we felt that based on the computational results, like there wasn't much reason to proceed with ProGen2. In terms of how these actual clusters were selected, it was as random as possible.
Oh, okay. Thank you. Thank you for answering all the questions. I think we go through all of them. Yeah, you can continue. Thank you.
Awesome. Cool. So yeah, now let's switch gears and start talking about alignment. So I want to get back to this point I mentioned towards the start of the presentation around model scaling and zero-shot fitness prediction. So as a community, we've largely decided that one of the ways we determine how good a PLM is based on how well its likelihoods, zero-shot, correlate with assay-labeled protein fitness data. But something that's pretty clear both for ProGen3, as we can see here, as well as all of these other families, is that the largest pre-trained models tend to have worse performance on this task. And this feels at odds with the idea that they're getting better and better at like learning these evolutionary distributions. And so in order to see like, are they still going to be useful for this sort of task? This is where we decided to reframe the problem of, instead of zero-shot fitness prediction, how good are they at few-shot fitness prediction? And in particular, like generating proteins that optimize certain notions of fitness that we care about, like using this limited amount of lab data. And so this is what brings me to the notion of language model alignment. So to draw some parallels, in NLP, language models are trained on massive scale web data, but they're often pretty challenging to use out of the box, and they can easily provide you toxic responses. And in order to use them as chatbots, you need to align them with human preferences. So let's say that I ask my chatbot, when did the first moon landing occur? A perfect answer would be July 16th, 1969. It's complete, it has the right date, no notes. Then an answer that's still pretty good would be just 1969. It's not as complete, but it's still correct, and it answers the question. An answer that's not so good would be 4th October, 1957. Well, this is a date. This is the date that Sputnik was launched, so it's just inaccurate. And then like a completely garbage response would be something like the sun orbits the Earth. Like one, not only is this completely unrelated, it's also just false. So to formalize how we do this sort of alignment, we're using a variant of direct preference optimization, or DPO. And I promise this is the only math on the slide, but let's go through it bit by bit. So in this notation, we have an assay X, and then you have a bunch of proteins in that assay, which are denoted Y1 through YN. And what you do is you consider all pairs of proteins, YW and YL. So here YW stands for Y winner and YL stands for Y loser. And the idea is you take all pairs where YW is better than YL, according to your assay. And what you do is you train this model P theta to increase the likelihood of your winners while decreasing the likelihood of your losers. So that's why you see this P theta of YW minus log P theta of YL in this equation. And you do this for every pair in your data set. Next, because you have this risk of catastrophic forgetting, you take your pre-trained model, pref, and basically taking this particular log likelihood ratio, you can show mathematically is a way to keep your new model, p theta, close to your pre-trained model, pref, in order to prevent some catastrophic forgetting. And in this particular setting, like X is our protein, or X is our assay, Y1 through YN, those are all of our proteins. And we say YW is better than YL if the fitness value measure for like YW is more than, let's say, like delta better than it is for YL. And like this delta, like the gap that you're setting is kind of case by case and depends on the noise level of your assay. Any questions, just because this is a foundational for the rest of the section?
So how different this formulation is from RLHF formulation?
So RLHF is an online reinforcement learning. You can show mathematically under certain assumptions that DPO, which is in this case, this is like a supervised offline formulation. You can show it is exactly equivalent to like one of the standard RLHF formulations, but it's a lot more tractable because it means you don't need to generate new sequences, which is a key component of RLHF. And in particular, you don't need a reward model. And with like a limited amount of data, it's just infeasible to train a good reward model.
Great, thanks.
Okay, so the first thing we use this for is a few-shot supervised prediction. And so here we take 8 protein gym assays that have at least 3 mutations from the wild type, and we create train test splits based on the number of mutations. So typically we'll be training on, for example, all of the single mutants, and then evaluating on all of the multi-mutation proteins. And so in this case, the train sets, they generally have like 500 to 2,000 sequences with at most 4 mutations from the wild type. And most of the test sets actually contain sequences with 10 or more mutations from the wild type. And so the blue lines here are the pre-trained model performance, both for ProGen2 and for ProGen3. And then in orange, I show what the performance of these models is after applying alignment. And so here, I guess the first takeaway is that alignment gives, and the way I'm measuring the fitness prediction performance is by the Spearman correlation between the model likelihoods and the fitness value of interest. So the first finding here is just that applying alignment period gives you a huge boost in performance. Second, larger models do better after alignment is applied. And third, the ProGen2 models respond better to alignment compared to the ProGen3 models. Sorry, other way around. The ProGen3 models do better after alignment compared to the ProGen2 models. And here I'm also comparing against this similar-ish method called confit, which works with encoder models and encodes a lot of like specific assumptions around the data being like substitutions only. And we see that the largest ProGen3 models outperform confit as well. Next, we align ProGen3 on this megascale thermostability dataset. And so this consists of 800,000 total proteins. And these are DMS assays of 479 different protein domains. And this consists of single substitutions, double substitutions, single insertions, and single deletions. So for the training, we train only on 90% of these assays on the single substitutions only. And for testing, we test on 10% of the assays across all mutation types. And in particular, the proteins in the test set come from different structural clusters from the train set. So these are like really distinct proteins, and it's measuring And this is measuring like a really, it's really measuring like if you learn a generalizable notion of protein stability. And what we find here is that, and here we're comparing against protein DPO, which is like a more specialized method, again, this is based on an inverse folding model. And so we find here that in terms of stability prediction for the full data set, larger models do better. ProGen3 does better than ProGen2. And the largest ProGen3 model across the full data set outperforms this protein DPO baseline, which is really specialized for the stability prediction task. What's really interesting is for these multi-substitutions, for the multi-substitution split specifically, protein DPO actually does really poorly, but alignment on the ProGen3 models especially does much, much better. And then, because again, these are generative models, we use them to optimize the protein stability of some of the proteins that we tested in the basically in that split GFP assay. So we take 32 of the wild types from that assay, and we basically prompt the model with the N-terminal or C-terminal 25% and let both the pre-trained and the aligned models generate the remainder of the sequence. And we see, according to Rosetta, the generations from the aligned models tend to be more stable based on like this delta REU. More details in the paper if you're curious. And then when we actually test these, in the lab, we find that for more prompts than not, the aligned models generate more stable or generate more highly expressing proteins, which, as we mentioned previously, for the split GFP assay, correlates well with protein stability. And then finally, this is some like relatively recent internal data that we got. I do have to anonymize it, but here we have some models that we aligned on internal data that we gathered over the past 2 years. Something like this was a few hundred different proteins measuring their activity. And so we aligned our models on that data. And then we essentially used Gibbs sampling to optimize those, to optimize like some of our best hits from prior rounds. To optimize like to introduce mutations that optimized our best hits from prior rounds. And so here in the dark lines, you see the performance of like each of these different seed sequences. And then in the light bars, you see how much improvement we got by using Gibbs sampling with these aligned models. And in some cases, you see like close to doubling in the performance of these proteins. And many of these proteins that were generated from this alignment round are much better than anything that we'd generated previously. Yeah, happy to take questions on the alignment section.
Oh, there's one question on the mutation prediction. How do you deal with deletions and insertions with using PLM?
Oh, it's with autoregressive models, it's actually really easy because you just take the likelihood like autoregressively and just like you don't need to do anything special. With encoder language models, it's definitely trickier. But for autoregressive models, like it's very easy.
I think there's another question. So basically it's asking in general, is scaling this much actually help generate more expressive proteins that align with the property? And seems like larger model generates some rare sequence out of distribution, but the expression of those might not be that great as expected.
Yes, but so I think that's all true in terms of like these unconditional generations. But I'll say that, so two things. First, if you're using these models for alignment, it's pretty clear based on these findings that like larger models respond better to alignment, both in terms of fitness prediction as well as some of the generation tasks. And second, like if you are in fact confronted with one of these really rare protein families, which like we've had to deal with a few of them in practice, like there were certain things that were impossible for us to design with smaller models just because those proteins were very out of distribution. But the larger models like already knew something about those families, which made them much more amenable to design.
Okay, cool. I think we've gone through all the questions.
Okay. Yeah. So yeah, with that, I guess time to wrap up. And so kind of getting back to this question, what does model scaling really buy you? So the two key benefits are first, you're more faithfully capturing the distribution of natural proteins, especially for rare families. And then second, you're getting better representations that improve your performance at downstream tasks, especially alignment with few-shot laboratory data. And so additionally, like given the gap between ProGen2 and ProGen3 across all of the evals I showed, better training distributions and model architectures also make a really big difference. In terms of what's next, we're continuing to train we're continuing to train our models on newer, larger versions of the PPA. As I mentioned at the start of the talk, PPA version 3, which is at this point already old, has 10 billion proteins, which I think is like 3 times larger than the next biggest data set out there. We're also exploring novel model architectures and training formulations. We're continuing to refine our alignment methods for real-world data sets like the slide I just presented. And we're continuing to battle test ProGen3 and other models in actual protein design campaigns. And with that, like we are actively hiring. I know a lot of people say that, but I promise we actually are. And this is across a wide range of roles, including protein design scientists who might have more of like a biophysics background, all the way to like ML scientists who maybe have a hybrid background or come from a more traditional ML background. But yeah, in terms of like, and yeah, like before I wrap up, I want to say that ProGen3 was truly a massive project that involved a lot of different teams across Profluent, like between the data engineering, the pre-training, the actual like split GFP experiment, all of the analysis for that experiment, and a ton of helpful discussions. All the people listed here helped a ton, and I had their faces on the slides where relevant. And yeah, like with all of that, thanks for your attention. I'm happy to take any last questions folks might have at this point.
Question here. Seems like models that are trained on distribution of natural sequences tend to mimic the distribution of functionality of those sequences, meaning the sequences you generate out of model will probably perform somewhere within mean of the distribution or not too far from the mean of the distribution of natural sequences, which obviously from design perspective is not what we want. We really want like edge of the distribution kind of generations. Does ProGen3 with the DPO as you described bring us closer there? And what else do you think is missing in the field to actually push closer to that edge of the distribution or even out of distribution performance?
Yeah, so I'll say like with this particular example, the baselines and like positive controls that we're comparing against, which like some of these seeds are already like outperforming some of those positive controls, those were based on like many rounds of directed evolution, which took like multiple years of design. And so essentially like in this single round, we were able to massively improve on those hits and really like get towards like more of this edge of distribution, like you're mentioning. So I think, alignment, DPO, and like, more sophisticated methods as they come out have a huge amount of promise for like pushing towards the edge of that distribution, which, just sampling from nature, whether that be like bioinformatically or through a model, probably won't be able to get you. In terms of like what's missing, I think there's a lot of different ways in which you can formulate DPO, or like, some other alignment methods. I think like online RL could be an interesting direction if you can come up with reward models that are robust enough that you believe them. There's also like just fundamental questions about like, how do you construct the best data set for model alignment? So I think all of these are like exciting directions.
Do you think a distillation of a model for a specific purpose, one of the ways?
What do you mean by that?
Distill the huge general purpose model into a smaller model for specific functionality.
I guess I'm not sure if distillation is necessary, because I guess distillation takes your big model and like, tries to encompass all of its capabilities in a smaller one. Whereas I think like actively using your supervised data in some manner like is more what I'm getting at here. I don't think they're mutually exclusive, but they feel like orthogonal directions.
We'll see what works.
Yeah. And maybe you align some distilled model and that works best. But yeah, I think they're not mutually exclusive.
I have a question here about the mutation effect prediction you did.
Mhm.
So I think from the result, you claim that the alignment helps a lot for the mutation effect prediction, but it seems that we still hadn't answered the question that you bring up before, that some larger model doesn't help the mutation effect prediction without alignment. Do you have any insight about this part?
Yeah, so I'll answer this in two parts. The first is, I think the fact that larger models are more responsive to alignment suggests that they are still learning like more useful representations that make them like more amenable to taking advantage of this supervised data. To your, I guess, big picture question, I think, yeah, like I've seen a couple of papers, which we also cite like in the ProGen3 work itself, that basically point to this idea that like, even if your evolutionary process is trying to optimize like some energy function, like let's say some protein fitness function, like you're gonna accumulate these random correlations just because like evolution itself is like, is an iterative process. You build up these correlations over time, and your observed distribution will not be like the exact distribution you would expect if you were just like sampling according to your energy function. That's one aspect. And then I think the other aspect is that often as a protein designer, the things we want our proteins to do are different from their natural function. And so if there is this disconnect between like what we think is protein fitness versus what nature actually selected for, then you can't really resolve, like there's just a tension that you can't really directly resolve.
I see. And I have a smaller question about the design of your experiment for the mutation effect as well. So you mentioned that there are eight arrays from the protein gene. So do you specifically train different models for different assays, or one model for all these assays? And when you select these assays, did they cover different fitness? Because we know that a protein gene contains different functions and fitness or stability, things like that.
Yep.
Have you ever tried like training on one task and evaluating on another task?
Yeah. So first question, it is one model per assay. Reason being that like each of the assays is dealing with like just a totally different protein family that has nothing to do with the others. I did do some initial experiments with protein gyms specifically. like where I was training on everything, it generally performed worse just because I think like it's just so diverse and contains so many different notions of protein fitness. More specifically with these 8 assays, they encompassed everything but stability. So for stability specifically, I used the megascale evals, but these 8 encompassed activity, organismal fitness, binding, and whatever the last one is in Protein Gym. I'm forgetting right now.
I see. Thank you. Thank you.
I see 2 more questions in the chat. Since you are, one question is from Rakesh. Since you're using experimental data, how does including this information into loss function via fine-tuning compare versus post-training alignment? And also, have you compared your model generations with family-specific MSAs along with simpler models?
So for the first one, like just doing SFT like works pretty poorly. There's just not enough data for SFT to work. And really, like with this DPO formulation, like the reason that I think it works so well is it is fundamentally a contrastive method, which gives you like n squared comparisons instead of just n samples. Sorry, could you repeat the second question? I couldn't quite hear it.
Second question is, have you compared your model generations with family-specific MSAs?
In what way?
I couldn't understand the question as well. Rakesh, are you available to unmute yourself?
Hi, yeah. So I was just asking, like, let's like if you take a family-specific MSA and use like a simpler model like variational autoencoder, right, or autoregressive autoencoder, and if you generate sequences, how does it compare it with the, let's say you use ProGen3, and like, do you get like similar kind of distributions, or in terms of does the data reproduce the same distributions, or does it look completely different?
Yeah, so we've done something similar where we've like essentially for Gibbs sampling, you can choose what function you're trying to optimize. So whether that be your aligned model's likelihood versus your pre-trained model's likelihood, the outcomes look totally different. Like if you're doing Gibbs sampling to optimize a pre-trained model's likelihood, like it doesn't look that good. Like it just looks, maybe a little bit better than your naturals. Whereas with like these really big like step changes, that's only really possible with like aligned models. In terms of what's in the paper itself, does this like this maybe sort of answers your question where like we're using the pre-trained model versus the aligned model to do like stabilization of existing proteins. And here we also see that generally the aligned model is outperforming the pre-trained model.
Okay. Yeah. So, so what I was looking, like generally I was trying to understand, like you're going in the larger direction, right? So is there a trade-off between like you're going the larger direction versus when you stick to family specific sequences and try to generate more around the natural space, does it help you generating faster rather than going larger and larger?
I guess like from our perspective, the cost bottleneck is not really generations, it's the experiments. And like the relative added cost of generation and scoring and stuff from these larger models, at this scale at least, is not enough for us to like, for it to override the like actual experimental costs. So I think like if we scaled up to say like 1 trillion parameters, which like maybe is or isn't a good idea. I think jury's still out. Like then we would start after thinking more seriously about like how expensive generation and stuff is. But like at the current magnitude, like the experimental costs are just so much higher that it kind of gets amortized out.
Okay. No, I was just curious, like in the natural language space, right? People are going towards the smaller language models now. So
They are.
is there a reason why we are seeing this way, but the NLP is going the other direction?
I think it's a question of scale because in NLP, like the large language models are multi-trillion parameter models. And like often smaller models are in this range of like 7 to 30 billion parameters, which is kind of where the ProGen3 work, like our largest models kind of fall in that kind of a range. So I think it's just like a question of like how large you're actually talking. Because I don't think anybody is really going all the way up to like 2 trillion parameters or something in proteins right now.
Thanks.
Cool, thank you. I think we'll go through all the chat. Sorry, I have one more question. So your model is basically using like an infilling pre-training objective, and there are also other models that have been coming up with like using like discrete diffusion score or protein language models. How do you see like the difference between these pre-training objectives, like which would be like better on the protein sequences? I guess for like traditional next token prediction, it might not work really well for protein. Because like protein is more like bidirectional information within them. So I was curious about what's your insight on this.
I mean, it's at some point it's kind of a practical question. Like if your model is using bidirectional attention, then masked diffusion makes more sense. If your model is using like autoregressive attention, then I think something like this fill in the middle objective makes more sense. Like just from a implementation perspective, like if you're using flash attention and you're doing anything, you can't really do anything that is not pure forward, like pure causal or pure bidirectional. Like if you have some sort of like weird hybrid thing, it just, yeah, like it just doesn't, like it's just not supported in flash attention. And so that was kind of like the practical reason we chose this implementation. I think we were curious about some of these like more hybrid methods, but it just wasn't practical to implement in a way that was fast.
Well, I think, thank you. Thank you. Sorry, we've been a little bit over time. And I just want to say, like, we really appreciate your great talk and answering all our questions. Thank you.
Thanks so much, everyone.
Thank you.
Thanks.